The overall goal of the following experiment is to assess the influence of pressure, shear stress, and external reinforcement on human intimal hyperplasia. This is achieved by simultaneous high-pressure perfusion of two segments from the same vein, while one segment is reinforced by an external mesh. The vein segments are then cut into sections for histological and molecular analysis. These analyses are used to evaluate the effect of an external reinforcement on intimal hyperplasia. The results show that an external reinforcement can reduce intimal hyperplasia that is normally observed after 7 days of perfusion.
Saphenous vein bypass graft is one of the treatments of choice in patients suffering from peripheral arterial disease. However, the development of intimal hyperplasia that leads to graft failure is the major limiting factor in such grafts. This remodeling is due to the adaptation of the vein to the arterial environment. To develop new therapies, the understanding of molecular mechanisms involved in this phenomenon is essential. Therefore, we have developed an ex vivo model of perfusion. One of the main advantages of our system is that it allows the study of hemodynamic parameters without any other confounding immune or endocrine factors. Alban Longchamp, an MD PhD student from our laboratory, will demonstrate the technique.